We're good? We're all on? Okay. Well, thank you for coming. For those of you who are here in the basement of Delia and Donald Baxter, and for those of you online, it's my pleasure to introduce Dr. Dawn Castor, who is an associate professor of medicine in the Division of Nephrology. Dr. Castor received her MD from UofL and completed a residency in internal medicine and was chief medical resident. Then she did clinical nephrology fellowship, followed by a research fellowship, and she is currently co-director of the Nephrology Research Center as of July last year. She's also director of the Glomerular Disease Clinic, and she's currently involved in 5 clinical trials. But she's also, importantly, the PI on an NIHR01, ABN1 dysfunction in lupus nephritis. And she's received a number of awards for her research. She serves as a reviewer for a variety of journals. She is an author on 42 peer-reviewed publications. And she's first author on a paper on treatment of primary IgA nephrology that was published in American Journal of Kidney Disease in February. So last month. She had 2 papers on which she was an author in December 2023, one in Lancet and one in the New England Journal of Medicine. And she's an author of 4 book chapters. And she's a runner. And I appreciate her involvement in talking with our second-year medical students for the NIDDK T35 grant each summer. So, Dr. Castor.
Thank you. Thanks for having me here today. This seems like an annual event, so I'm happy that I keep getting invited back to talk about lupus nephritis. Today I'll be talking about the utility of biomarkers in lupus nephritis because one thing that kind of links most of my translational research is it is all under the umbrella of biomarker research. Which it's easy to do because biomarker is such a broad term, and we'll be talking about that shortly, because it applies to a lot of areas of medicine, not just nephrology or autoimmune diseases. And I do recognize I'm here at the Molecular Endocrine Grand Round, so I hope to bring in some aspects that may relate to endocrinology in this talk. So I do have some disclosures. I am a paid consultant and on the Speakers Bureau for some pharmaceutical companies. I'm also PI on industry-sponsored clinical trials, and I have funding through the NIH. None of those will be affecting the content of the talk this afternoon. So learning objectives. So we're required to put together a variety of learning objectives, and I think it's a good way to summarize the main points that I'd like you to get out of the talk. Be able to identify current clinical biomarkers that are used in lupus nephritis. The learner will be able to identify limitations of these clinical biomarkers, and I think when it applies to any complex disease, we do have a limit in the biomarkers we have in the clinic. And then understand the potential role of novel biomarkers in this field. I throw this up here because one of my colleagues who's much younger than me, I've now accepted that I'm mid-career. One of our junior faculty members brought up House as an inspiration for him going into medicine, and I was already in medicine when the show was on board, but he's very known for saying it's not lupus. So I don't know if you guys are familiar with House, but in my world, it's always lupus. Well, maybe not always, but a lot of the time it is lupus. So about 30% of the patients I care for in the glomerular disease clinic are patients with lupus nephritis. So what is lupus as a whole? Of course, I'm most interested when it affects the kidney, but it is a complex disease affected by genetic, environmental, immunoregulatory, hormonal, and epigenetic factors. that lead to loss of self-tolerance and the development of autoantibodies, immune complexes that then activate an immune response, which includes B cells, T cells, innate immune cells, cytokines, that all lead to end organ damage, affecting kidney, skin, lungs, the brain, and the cardiovascular system. Something really interesting, we know that most autoimmune diseases affect women more than men. But lupus, among those diseases, really has a disproportional amount of women affected. So 9 out of 10 patients with systemic lupus are women. So there's a huge hormonal role. And to bring this into the endocrine field, I thought it's interesting. This isn't my area of expertise, but there are known sex-based differences in immune responses. And many of these really are most apparent during puberty. So especially, and it makes sense because lupus is a disease that's usually diagnosed in women of childbearing age. So beginning in adolescence through 20s and 30s. So typically young women. In women in post-puberty and adulthood have increased levels of immunoglobulins, increased levels of B cells. Males have increased T regulatory cells, which actually help regulate the immune response. And pretty much across the board, most immune responses are increased in women, with a few exceptions. So I mentioned this already, but lupus is disproportionately affects women and minority populations as well. So 9 out of 10 patients with systemic lupus are women. Patients with African and Hispanic ancestry are more likely to be diagnosed with lupus, and specifically more likely to have lupus nephritis. And patients with African and Hispanic ancestry are more likely to develop end-stage kidney disease from lupus nephritis. About 50% of patients with lupus develop lupus nephritis, and up to 30% of patients with lupus nephritis will progress to end-stage kidney disease, and complete remission occurs less than half the time with standard of care therapies. So we really have some health disparities and a significant disease burden in this population. So that's where biomarkers may play a role. And there have been a lot of studies on biomarkers. So I just did this PubMed search on lupus biomarkers, not even specifying lupus nephritis. And there have been over 8,000 publications to date on lupus biomarkers, and then almost 2,000 on lupus nephritis biomarkers. So quite a few publications out there. However, to date, we don't have any novel biomarkers that I can really order in clinic. So what is a biomarker? And I think it's really important that we define this because we talk about this in the setting of a lot of diseases. Of course, I'm a nephrologist with a focus on lupus nephritis, but many of you in the room may be trainees or have a focus on endocrine, and I'm sure you have your list of novel biomarkers and novel biomarker research. But a biomarker is simply a characteristic that is objectively measured and evaluated as an indicator of either a normal biologic process, a pathogenic process, or a response to therapeutic intervention. So every day when we see patients, we're using biomarkers. Weight is a biomarker. BMI is a biomarker. Blood pressure is a biomarker. A1C is a biomarker. So a lot of the clinical tests are biomarkers, but we tend to think of novel biomarkers and research as new things that can allow us to really personalize medicine. So the traditional biomarkers that I test for in my clinic patients include proteinuria. And proteinuria can be measured as a urine protein to creatinine ratio or a 24-hour urine collection. Most patients don't like carrying around the 24-hour jug, so we may get that on occasion, but usually we're getting a random sample. Creatinine with a estimated GFR to see how well the kidney is filtering, hematuria and urine sediment evaluation. Then these autoantibodies. So, of course, anti-nuclear antibodies are present in all patients with lupus. Anti-double-stranded DNA antibodies are more specific for lupus nephritis, although they're not perfect. I have several patients that have negative anti-double-stranded DNA antibodies, but still have lupus nephritis. So there are other antibodies that probably are involved. Anti-C1q antibodies also thought to be more specific, but a little less sensitive for lupus nephritis. I apologize. I'm going to turn this off. And complement levels tend to be low because there's a complement activation and deposition into the kidney tissue. But again, not a perfect biomarker. And then the kidney biopsy, which is the gold standard for diagnosis of lupus nephritis, and then the classification. So I want to talk first about proteinuria because it is currently being used as a marker for response in many clinical trials. And it is looked at as, one of the best biomarkers we have for prediction of long-term outcomes in patients with lupus nephritis. So these are two large trials, primarily in the European population that had long-term data. So the MAINTAIN trial had 10-year follow-up data, and the Euro Lupus Nephritis trial had 7-year follow-up data. And they went back and they looked at proteinuria at baseline compared to month 3, month 6, and month 12. And they found while baseline proteinuria was similar in the groups with good long-term outcome here in black, and in the light gray, poor long-term outcome, you can see an early separation even at month 3 of proteinuria decreasing in the patients with a good long-term outcome. And by month 12, you had a really good prediction of who was going to have the best long-term outcome. So the patients whose proteinuria didn't fall, at least very much from baseline, did the worst. And they found that a proteinuria level corresponding to about 0.7 grams a day had the best sensitivity and specificity at predicting the 10-year outcome. Similarly, in the Euro Lupus trial, looking at 7-year outcome, they looked at 12-month proteinuria between 0.7 and 0.8 grams, having the best specificity and sensitivity at predicting long-term outcome. So we know that proteinuria 12 months after diagnosis, so is a good marker of response to treatment, and thus the response to long-term outcomes in patients with lupus nephritis. Those studies were done in Europe, so their population isn't quite as diverse as it is here in the United States. And there was a similar study in a more ethnically diverse cohort that found very similar findings. This is looking at 7-year follow-up, and they found a cutoff of around 0.8 grams a day of proteinuria was the most sensitive and specific for long-term outcome in patients with lupus nephritis. And interestingly, proteinuria at 1 year was a better predictor than some of the other potential confounding factors such as gender, race, anti-double-stranded DNA levels, and histologic class. Creatinine, we think, obviously, the lower your creatinine is, you should do better. But interestingly, it doesn't add a whole lot to the 10-year outcome. So proteinuria of less than 0.7, this is looking at the MAINTAIN trial, had a sensitivity of 71%, specificity of 75%, and a positive predictive value of 94% of good long-term outcomes. You add in serum creatinine of less than 1, and you get a little bit more specificity, but not a huge change. And interesting, adding urine sediment to that did not give any better prediction. So unfortunately, proteinuria is not perfect. We still measure it all the time because it is a good indicator of response to therapy. But proteinuria and traditional biomarkers can be discordant with disease activity. So this study looked at patients who had achieved clinical renal remission based on proteinuria levels over here. And they did biopsies in all these patients. While the vast majority of them had low activity levels on biopsy, some of these patients that were in clinical remission still had a high activity index. Without going into the details, an activity of index of greater than 5 is still indicative of a high level of inflammation on a biopsy in a patient with lupus nephritis. So they found that about a third, so 29% of patients who had no proteinuria but had a repeat biopsy still had high histologic activity on the biopsy. Conversely, because these biopsies were done serially in these patients, there were patients that had proteinuria above 500 milligrams, which is a typical cutoff for remission, and they had a complete histologic remission, but still had persistent clinical disease based on the level of proteinuria. So 62% of patients with complete histologic remission still had persistent proteinuria. So this group over here with a low level of proteinuria but a high activity index on biopsy represents a group of patients that may be inappropriately taken off of immunosuppressive therapy because clinically they seem stable, but they still have activity. And this group over here represents patients that may inappropriately be continued on immunosuppressive therapy despite having no activity because they have persistent proteinuria. So certainly the immunosuppressive treatment that we use to treat lupus nephritis is not without side effects and risk. So better identifying patients with disease activity becomes important. So this study actually took the previous study a step further. So they wanted to see what happened if you were to do these protocol biopsies in patients and withdraw immunosuppressive therapy when blinded to that second biopsy. So essentially, they took 36 patients that were in complete remission for 12 months that had received at least 3 years of immunosuppression therapy. And patients underwent a second biopsy and then tapered off immunosuppressive therapy and were followed for 24 months for flares. And they found that a flare occurred in 11 of the patients that were taken off immunosuppression. And when they went back and they looked at the second biopsy, the protocol biopsy that was done before withdrawing immunosuppression, they found that the activity index of patients who experience LN flares was higher in the patients that had flares for the most part versus those that had no flare. So the patients that had no flare all had an activity index of 2 or lower, where the majority of patients with flares were above 3. There were a few, one that had a flare that had an activity index of 0, but for the most part, they had higher levels of activity, which suggests that we need a better biomarker than our typical proteinuria and stable creatinine to decide whether or not to take people off immunosuppression. So another limitation is that while proteinuria is a good predictor of long-term outcomes, low-grade proteinuria doesn't exclude significant kidney injury in some patients with lupus nephritis. So this is looking at patients at a single center over a 10-year period. And, typically we think about less than 0.5 grams a day as not being significant. But about 20% of patients in this study had proteinuria of less than 0.5 grams a day and still had significant findings. So 85% of patients with less than 0.5 grams a day and 76% of patients with proteinuria of less than 0.25 grams a day had a class 3 or class 4 proliferative lupus nephritis lesion. And only 11% had a class 2 lesion. So, and that's the less than 0.5. So you can still have significant disease despite low levels of proteinuria. Now, the caveat to this is these patients were probably biopsied for a reason. They might have had increasing creatinine levels or hematuria, low complement levels, but proteinuria is not the perfect biomarker that we were hoping it would be. This is another much larger study. So the Accelerating Medicines Partnership is a consortium developed by the NIH to study rheumatoid arthritis and systemic lupus, utilizing a network of multiple centers. And they found, looking at the AMP partnership patients, 20% of patients who underwent kidney biopsy in the AMP SLE program had a UPCR of less than 1. And patients with a UPCR of less than 1 had a median activity index of 4.5 and chronicity index of 3. So they still had quite a high level of activity. Interestingly, some of the other biomarkers we frequently look at, like double-stranded DNA levels, low complement levels, did not distinguish well between the classes with UPCR of less than 1. So that brings me to what is the ideal biomarker for lupus nephritis? So it would, 1, identify those at risk for developing disease, 2, establish the diagnosis and distinguish between active and chronic disease, 3, guide choice and duration of therapy, and 4, be reproducible in a commercially available assay at a reasonable cost. So obviously it's not going to be just one. So there won't be a single ideal LN biomarker, but probably several that will help lead to a more personalized approach to therapy in patients with lupus nephritis. And it will be likely a combination of genetic biomarkers that identify patients at risk of disease, urinary biomarkers outside of proteinuria that help identify disease activity, serum and blood biomarkers looking at novel autoantibodies that help capture patients that don't have our traditional autoantibodies, and then tissue markers that help identify immune changes in the kidney. So I'd like to start with genetics, because this is where my research started. And this is just this comes from a review by George Sokos that came out a little over a decade ago, but I really like how he highlights the diversity of genes associated with lupus and separates it nicely by immune responses. So you can see a host of immune responses are involved in the pathogenesis of patients with lupus nephritis. And it's likely that a different immune response may exist in different patients. So dendritic cell functioning, interferon functioning, the immune complex processing, T cell signaling, cell cycle, apoptosis, cellular metabolism, B cell, and then transcriptional regulation, as well as some other genes have been associated with development of lupus. And this is all lupus, not just lupus nephritis. I'd like to focus on this dendritic cell function and interferon signaling because the gene that I've studied personally is TNIP1. And this was work that I did during my research fellowship. And ABN1, just to confuse you, is the protein that TNIP1 codes for. So TNIP1 is the gene and ABN1 is the protein. And we found that ABN1 dysfunction in an animal model led to lupus-like autoimmunity. And we were interested, since these TNIP1 mutations had been identified as risk factors for the development of lupus, to see if there were associations specifically for lupus nephritis. And we identified two genes, one in African Americans and one in patients with European ancestry, that increase the risk not only for lupus, but the risk for lupus nephritis. And there are several genes that have been associated with lupus nephritis on top of lupus. So in high-risk patients, so patients that are already identified as having SLE, it would be nice to be able to identify if they have any of these genes, which also increase the risk of lupus nephritis, which may lead to earlier detection of their nephritis. So these genes that are associated with increased risk of lupus nephritis in patients with lupus involve both the innate and adaptive immune system, because both are important in the pathogenesis of lupus, but also include intrarenal genes such as ApoL1, which is a known risk factor for development of end-stage kidney disease from hypertension, as well as lupus nephritis and in African Americans. And interestingly, ApoL1 increases your risk of kidney involvement in a number of other diseases except for diabetes, which we don't quite understand why, but and this gene is more common in patients with African ancestry because the gene variants actually were evolved because they protected against African sleeping sickness. Some other genes involved: programmed cell death and immune complex clearance, because the clearance of immune complexes are implicated in the development of lupus. You get that initial response by poor immune clearance of apoptotic bodies, leading to the anti-nuclear antibodies that initially form. So what is the application? So I mentioned one, which is identifying those at risk for developing disease, but also understanding the immune responses may be different in an individual patient based on their genetics may lead to a more personalized approach to medicine. Now, I'm not suggesting any of these individual therapies. Many of them have been approved for other disease processes, but in the age of biologic therapies where you have different immune targets, you could see where identifying which pathways are upregulated in individual patients based on their genetic background may lead to a more precision medicine approach to therapy. And this came from a review of genetics in lupus nephritis by Monroe and James. And again, the caveat is these would all need to be studied further, but you could see where the potential gene identifying the aberrant immune response could be linked to a potential therapy. So I'd like to move on to urine. As a nephrologist, I love urine. Every time I go into a patient's room, the first thing I look for is the bag. And if they're making urine, then I get excited when I see urine. So it's a sign that the kidneys are at least working somewhat. So there are several novel urine biomarkers in lupus nephritis. And one of the reasons we look at urine is because it kind of is our low-hanging fruit of what's going on in the kidney. So we can't do repeat biopsies on everyone. Biopsies are invasive, and not all patients want to be subjected, and understandably, to serial biopsies. So the next best thing is looking at urine because it gives us a direct idea of, some of the inflammatory cytokines that may be coming from the kidney tissue. So there's several novel urine biomarkers that have been looked at in lupus nephritis. This is not a complete list. As I mentioned, there are over 2,000 papers. But I wanted to put together a table based on the ones that are most promising, that have been correlated with disease activity, studied longitudinally, prediction of flares, or can correlate with histology or biopsy class. And I've highlighted one here because it's one that we studied in our lab, and it's interferon gamma-induced protein 10, or IP10. And we found in our local cohort of lupus nephritis patients that both serum and urine levels were higher in patients with lupus nephritis versus controls. We also found that patients with proliferative lupus nephritis had the highest levels. So proliferative is the class 3 or class 4 versus membranous, which is class 5. We tried to pair patients, or we did pair patients, that samples. So each of these dots represent a patient that had active disease, and that same patient going into inactive disease. And the vast majority of patients, IP10 levels went down from active to inactive, but there were a few outliers here. And we looked at these closely, and we found that one of those outliers had a flare like a month later. And how did we identify active disease versus inactive disease? Well, we used proteinuria, which I already told you was imperfect. So we really do think that this does represent disease activity. There's definitely a trend there. And we also found that when looking at serum levels of IP10 versus urine levels of IP10, that compared to other glomerular diseases, lupus nephritis patients had higher levels, but glomerular disease patients also had higher levels than controls in their serum and the urine. These are glomerular disease controls here. And then finally, we wanted to distinguish between membranous lupus nephritis and membranous nephropathy. And we found that there wasn't a difference in serum levels of IP10, but there was a difference in urinary levels of IP10. So this is a potential future biomarker in lupus nephritis with urine levels being more indicative of active lupus nephritis than the serum levels. I'm bringing up another study by the AMP program, the Accelerating Medicines Partnership. And this was a study that enrolled 225 SLE patients that had were going to get a kidney biopsy, and they wanted to look at the correlation of the kidney biopsy as well as urine biomarkers in this group of patients, and identify correlations with both the biopsy activity and look at longitudinal molecular signatures and histologic phenotypes of response groups. So they wanted to look at if they could identify anything that correlated with both activity and treatment response. So pretty extensive study. And I'm not going to go into all of their results, but I thought it was really interesting that they did a pathway analysis of the urinary biomarkers, and they indicated that neutrophil degranulation is a key pathway in active lupus nephritis. And one of the reasons I found that interesting is because we were already studying neutrophil granule proteins as a urinary biomarker in lupus nephritis. And I always get excited when another group shows work that kind of helps reflect the work that we're doing, because it makes me think I'm on the right path. And I wanted to bring back, because we are in endocrine grand rounds, this idea of sex-based differences in immune response, specifically in neutrophil responses, because that's an area of focus in our lab. And this group, led by this paper by Gupta et al., looked at changes in female neutrophil responses versus male neutrophil responses, and they showed that the female human neutrophils, and these are just healthy controls, so not patients with lupus, had increased interferon signatures. So interferon responses and innate immune responses are important in the development of lupus nephritis, and perhaps part of that immune risk is already there, even in healthy individuals, with females expressing much higher interferon signatures in their neutrophils. So this isn't an area that I specifically study, but I'd be happy to collaborate if anyone has any ideas on ways to look at sex-based immune responses and how they may correlate to what we're finding in our lupus nephritis patients. The hard part is I don't have very many male lupus nephritis patients. I think we only have a couple in our cohort. So it's really hard to find disease controls that are males. So we looked at neutrophil degranulation proteins in patients' urine, and this is a volcano plot looking at several of these proteins. And this is the fold change, and these are the ones that were significant on our first round of testing. Of course, each of these needed to be validated independently. And of these markers, alpha-1 AG was consistently elevated in lupus nephritis urine, and also corresponded to activity. So this is looking at healthy donors. This is lupus nephritis class 3 and 4, so the proliferative forms of lupus nephritis. This is class 5, pure class 5, so just membranous. This is looking at other disease control, so minimal change disease and membranous glomerulonephritis. And we looked at these because on our manuscript that was submitted, some of the reviewers were concerned that the proteins we were seeing was just because these patients had high levels of protein, and this was just representing proteins that were, in the bloodstream. We all have a high number of neutrophils, right? That were kind of leaking out into the urine indiscriminately. I can guarantee you these patients with minimal change disease, they were nephrotic. So we made sure we had significant disease controls, and you can see It does not seem to be an indiscriminate protein excretion here. So we were really excited about these results. And this is looking at active versus inactive. So these are impaired patient samples. And this is an ROC curve looking at the predictability of alpha-1 AG as a predictor of lupus nephritis activity. And this data, I'm excited, is also going to be presented by one of our summer students, Lashaya Davis, who's going to be presenting it in New Orleans here in a couple days. So she did some of this work this last summer as a T35 student, and also came into the lab to get some additional data on her spare time as a medical student. So, you know, we're very excited that she's part of that. This is some additional work from that project showing that patients with lupus nephritis have enhanced degranulation of neutrophils, and that the serum from lupus nephritis patients also stimulates degranulation in healthy donor neutrophils. And I'm not going to go into a lot of detail here, but CD66B is a marker of specific granules, and FMLF is a basically stimulates neutrophils to degranulate. So that's kind of our positive control. And you can see over here in black, patients with lupus nephritis actually have higher basal degranulation, so higher activity at baseline compared to controls. And they also have increased response to FMLF. This is looking this is looking at the patient's neutrophils and healthy donor neutrophils, but we wanted to look to see if there were substances from the serum of patients with lupus nephritis that also activated neutrophils, and we were able to see an increased response. So moving on, I want to make sure I finish on time. I think this is, there's a lot to go over. Looking at plasma and serum markers and tissue markers, there are several different markers that can play a role, including autoantibodies, cytokines, chemokines, adhesion molecules, damage markers, microRNAs, and immune signatures. We've done some work in looking at autoantibodies to glomerular antigens. And this is work that I did early in my career as part of a K08 Career Development Award. And we identified two glomerular proteins, annexin A2 and mosin, as targets of autoantibodies in patients with proliferative lupus nephritis. And this is looking at normal controls, lupus controls, so patients with lupus and without nephritis, class 5 or membranous lupus nephritis, and proliferative, which includes class 3 and class 4 lupus nephritis. And they had increased circulating autoantibodies to annexin A2, and they had increased circulating autoantibodies to mosin. With mosin, we also found increased glomerular staining, especially around the capillary loops, in patients with proliferative lupus nephritis versus membranous, and then we had some control glomeruli as well. Autoantibodies can also be targeting specific glomerular proteins, leading to the development of lupus nephritis. I'd also like to discuss tissue immune signatures, because this is an area that I find, exciting, because it could help identify patients that may respond to certain interventions. So this group looked at type 1 interferon and neutrophil transcripts in patients with proliferative nephritis, and found an increase in patients with, so they looked at proliferative and membranous, and they found increased transcripts of interferon in patients with the proliferative as compared to the membranous, as well as this is defensin, which is a neutrophil granule protein, suggesting that these pathways may be potentially good targets for future therapies. And I don't have a lot of time to talk about future therapies, but there is a FDA-approved therapy for lupus, not lupus nephritis, called anifrolumab, which targets interferon, and is being studied right now. We're actually part of the phase 3 trial. We're looking at it as add-on therapy for patients with lupus nephritis. So adding it on to standard of care to see if it helps get more of an immune response. And you can imagine, while we're not looking at these immune signatures in the patients in their biopsies, it would be interesting to also look at the molecular level to see if the patients that did have a benefit had higher levels of interferon proteins or interferon mediated proteins in their biopsies. So we're not quite there yet, but we are looking at these new pathways for interventions. So in conclusion, because I want to make sure I finish with time to spare, there's not a single biomarker that is the end-all be-all for lupus nephritis. It's going to involve multiple biomarkers. But hopefully we'd be able to identify those at risk for developing disease, establish diagnosis, identify flare, and distinguish between active and chronic disease, help guide choice and duration of therapy in the future. And as we have more and more therapies on the horizon, I think it's going to be really critical to have better biomarkers to guide choice and duration of therapy. So in a way, I feel like industry, because we're developing all these new biologics that target different pathways, it's going to be important to find out which pathway may be activated in each individual patient to decide which biologic would be ideal to use. So, right now, and this talk was not on therapy, but right now we kind of blast the immune system with steroids, often causing steroid-induced diabetes and all these other bad side effects. And in addition to steroids, using kind of broad-based immunosuppressive therapies like cyclophosphamide or mycophenolate. But now we're entering an era where there's novel B-cell therapies, novel T-cell therapies, potentially interferon-based therapies. So I think it's really going to be critical to identify which patient may be more ideal to respond to which add-on therapy to decide to use. And with that, it's a big group of people to acknowledge. So obviously research doesn't happen in a vacuum. All that work was the work of many. So I'd like to highlight my colleagues at UofL that have been a big part of my research career. We've been very fortunate to have some wonderful medical students rotating through the lab, and I highlighted these two here because they were part of the T35 summer program, and some of our outside collaborators as well. And I want to make sure I give everyone the event code, because I know that's important, but I'll take any questions while everyone's writing that down. Okay. Oh, great.
So the degradation of the neutrophils, is that something systemic, or do you think it takes place in the kidney and the markers are both in the serum and in the urine? And
That's
I don't really know anything.
Yeah. Yeah. So that's a great question. So we think that patients with lupus have neutrophils that are kind of primed for degranulation. So they're already kind of ready to degranulate. But we do think that the degranulation in the kidney is causing direct tissue damage, and is a marker of disease activity. And if you actually look at some of the biopsy specimens, you can see, so neutrophils are a big, they're the most abundant immune cell in circulation. So we see a lot of them. And if you were to take normal kidneys, you would occasionally see a neutrophil within the capillary lumen of a glomerulus. But we see migration into the tissue, and then that migration goes from the tissue into the urinary space, and we don't see that in normal. So we think that the urine degranulation markers are evidence of inflammation in the glomerulus. They're all just in research, I know. So the question is, are any of these commercially available? Other than the one that I put on that slide of our traditional biomarkers, even the ones that have been around for a decade, like IP10, for example, has been studied by other groups, not just ours. It's not available. At this time, there's no novel urinary biomarker panel. Part of that is because everybody's using their own assay. There's no kind of uniform assay that's been validated across cohorts. I'm hopeful that the AMP program will lead to more of that because they have multiple centers and a more varied population. And part of the goal of that program was to have better biomarkers to help identify patients with disease activity, but also identify immune response. So we'll see in the next few years. I hope something will be out there, but at this point, there's no novel biomarker commercially available in lupus nephritis. There's a need for it.
There are questions in the chat box.
Chat. Oh, there's questions in the chat. Sorry. How do I find that? Sorry. So do I end slideshow?
Yeah.
Okay. I apologize. I'm looking. I might need some help. Okay. Oh, stop sharing. And then chat. Oh, I just have to. Now I can see. Okay. Oh, great. All right. So microRNAs are some, and I don't have that as part of this talk. I'm happy to get back to you on the question of microRNAs. Can we use complement, opsonins, along with other symptoms? It is true that some patients, so sorry, I'm trying to figure out how to answer. I might have, these are tough questions. I might have to get back to you on that one. Definitely impairment in the complement system has been associated with a genetic increased risk of lupus because of poor clearance. So complement has kind of a dual-sided role in lupus nephritis. We need complement to be able to clear immune complexes, and we need complement to be able to, but on the flip side, complement is definitely activated in patients with lupus nephritis. It's a big challenge, yes. Is there a compound of natural origin tested against SLE? Another good question. So for SLE treatment, here in the United States, I don't think anyone's tested any natural compounds for the treatment, but I know in Chinese medicine, there have been some anti-inflammatory compounds that have been looked at, certainly in animal models. There have been some anti-inflammatory compounds looked at for SLE, but nothing that's FDA approved or has a large clinical trial. So I can't tell you to go get a specific natural compound to treat lupus. I hope I addressed some of those. I'm happy to look into a couple of the other ones and get back to you. Anything else? Okay. Thank you. I appreciate your all's time. I know it's late in the day, so thank you for sharing your afternoon with me.